Hello, everyone. I am Dr. Vinod Shinde. In the previous lectures, we have discussed about EMP pathway, HMP pathway, and ED pathway. EMP pathway was run by majority of microorganisms. HMP was a shunt where EMP pathway is absent or some enzymes of EMP pathway are absent. HMP was run. ED pathway is observed in Pseudomonas genus and so now we will discuss about the phosphoketolase pathway. The heterofermentative lactobacilli and bifidobacteria possesses phosphoketolase pathway, which is a variation of HMP pathway. Keep in mind that it is a variation of HMP pathway. There are two types of phosphoketolase pathway, that is pentose phosphoketolase pathway and hexose phosphoketolase pathway. Pentose phosphoketolase pathway was run by Leuconostoc species, Leuconostoc plantarum, Leuconostoc mesenteroides. And the species is lacking some enzymes or is lacking the pathway, that is EMP, HMP, and ED. The carbon source is ribose as well as other pentoses are observed in the pentose phosphoketolase pathway. Pentose 5-phosphate and xylulose 5-phosphate are formed via the HMP pathway. It is the variation in HMP pathway. So it was found in Leuconostoc mesenteroides and Leuconostoc plantarum species, in which EMP, HMP, and ED pathway are absent. The ribose, xylose, and arabinose sugars are used in this pathway. That is, pentose sugars are all used in this pathway. Ribose is all three sugars can be utilized by this organism in different ways to produce a similar product, and we are depicting the same thing, that is ribose is converted into ribose 5 phosphate by ribokinase enzyme utilizing one ATP. Ribose 5 phosphate is converted into ribulose 5 phosphate by ribose phosphate isomerase enzyme. Ribulose 5 phosphate is reversibly converted into xylulose 5 phosphate using ribulose phosphate epimerase enzyme. Xylulose 5 phosphate splits into acetyl phosphate and glyceraldehyde 3 phosphate, which of which acetyl phosphate gives you acetate by acetokinase enzyme, and glyceraldehyde 3 phosphate goes to EMP pathway producing lactate finally, that is lactic acid. The splitting of xylulose 5 phosphate into acetyl phosphate and glyceraldehyde 3 phosphate was done by phosphoketolase enzyme. The second sugar, that is ribose sugar used, is xylose. Xylose was converted into xylulose by xylose isomerase enzyme. Xylulose was phosphorylated and give ATP was utilized here to give you xylulose 5 phosphate, which was split by phosphoketolase enzyme into acetyl phosphate and glyceraldehyde 3 phosphate. Acetyl phosphate gives you acetate, glyceraldehyde 3 phosphate gives you lactate through EMP pathway. The third pentose sugar used is arabinose, where arabinose is in the presence of arabinose isomerase gives you ribulose, which was reversible reaction. Ribulose in the presence of ribokinase enzyme utilizes one ATP to give you ribulose 5 phosphate, which was converted reversibly in the presence of ribulose phosphate epimerase enzyme to give you xylulose 5 phosphate. The ultimate xylulose 5 phosphate was split into acetyl phosphate and glyceraldehyde 3 phosphate by using enzyme phosphoketolase. The acetyl phosphate reacts further with acetokinase to produce acetate, and glyceraldehyde 3 phosphate goes to EMP pathway, giving lactate in the presence of oxygen. It goes to pyruvate, and pyruvate converts into lactic acid or lactate. This pathway was run by the species of Leuconostoc, that is Leuconostoc mesenteroides and Leuconostoc plantarum, in which EMP, HMP, and ED pathway was absent, and it utilizes only pentose sugars. The next pathway is hexose phosphoketolase pathway, which we will discuss in the next lecture.